So let me say thank you very much to the organisers for inviting me. It's a real pleasure to come back to ISMB. I've not been for several years. And however, that said, I really bit off more than I can chew here. 50 years of protein structure bioinformatics is so big, and trying to give an overview of it, I think I will fail spectacularly. Can you hear me? I'll stand here. It's all right, I'll stand here. Yeah. So I wanted to put this in perspective because I think we forget. You know, we've just heard the brilliant talk from Martin Steininger, and things have moved on so much. But 50 years ago, we hardly knew anything. So the first protein sequence wasn't determined until 70 years ago. The first structure, 60 years ago. Margaret Dayhoff started her collection of sequences in 65. But then many protein sequences were characterized. You can see here the pink book of protein sequences. And since then, we've had proteins and structures galore. And of course, we've been really blessed because we have I think the oldest biomolecular data resource from all the different aspects of biology. And the Protein Data Bank was set up in 1971. And I have to say that all of my work has absolutely depended on this database and the structures that have been deposited therein. And of course, that database has grown enormously. Actually, this is a week old, so now it'll be sort of 219,000 proteins that are in the PDB. I started looking at proteins really in 1973, almost exactly 50 years ago, and there were 20 protein structures in the PDB. I was in charge of getting the magnetic tape, 12-inch magnetic tape, and putting it on the system. And so I've been involved with the PDB for many, many years. And of course, the thing that drove me and drove many people in the field was to try to predict the protein structure from the protein sequence. And many of the people in the field were physicists, not biologists, like I was, and trying this very simple problem, take a string, a polypeptide chain, and fold it into 3 dimensions. It's a very easy problem to understand, but my goodness me, it was a difficult task for everybody to look at. And so I'll go through some of the main developments. So when we started off in the 70s, the idea was to sort of tease apart the structures. So we were looking for basic principles of structure, such as which amino acids like to be in a helix, what's the phi-psi values for the different amino acids, what were their distribution on the Ramachandran plot. We looked at the hydrophobic core, we looked at hydrogen bonds, we looked at disulfide bridges, and we looked at all the different characteristics of secondary structures that are, of course, the sort of basis of the 3D structure. So looking at finding out that helices were actually bent. For me, I was quite surprised. I didn't think they'd be bent. And they clearly are because of the water interactions on one side. The twist of the strands, the turns and the residue preferences of the turns. We spent hours looking at term preferences, etc., for the different amino acids. And also we were looking at chirality because we were getting new structures. And it turned out that the beta-alpha-beta unit, where the beta strands are parallel, is almost always right-handed. I mean, it has to be the best rule, if you want a rule, in protein structure, because about 99% of those beta-alpha-beta units are right-handed. And it all comes down to this little difference between a D and an L amino acid. And I find that still fascinating, that one difference in where you put the C beta changes not only that you get a right-handed helix instead of a left-handed one, but the whole topology is affected by that. And obviously also solvent accessibility was very important in looking at that. So we looked at these turns and bulges and hairpin loops, and we actually produced a book. My student, Just Singh, went off and produced a book of the 20 side chain interactions. It has to be probably the most boring book, but it was quite interesting because you could see what was going on. You could understand it. It was very much doing these analyses to understand why a certain sequence led to a type 1 beta turn or a type 2 beta turn, whatever it happened to be. But of course now, all of this is very easy because you can characterize it, you can pull these motifs out from the PDB very easily. There was also the problem of actually visualizing the structures, because I spent hours, and the older people in the audience will remember looking down the 3D glasses to work out whether a beta alpha beta unit was right-handed or left-handed. And it's really not that easy when you're looking down in a 3D glasses. And so the development of new methods to visualize these structures was really important. And so, you know, the original, Irving Geis did the original picture of the hemoglobin. And then Jane Richardson's ribbon diagrams came along, which she drew by hand. I mean, this was a phenomenal achievement to draw these. And I went to visit her, and she had all these pages with, and she spent hours looking at one structure to get the right thing. And of course, that was then reproduced automatically. CABS produced an algorithm that drew them for us on the computer. And that made a huge difference, because now you could look at the fold of the protein much more easily. We developed topology diagrams, Lig plots to look at how ligands interact, hydrogen bond diagrams. And of course, David Goodsell went a step further and represented these proteins in the cell and showed how crowded it all was. So these different ways of visualizing what's going on, to me, was very important because it allowed us to understand some of those principles. So the early tools, really, at the beginning, it was all about predicting the secondary structure accurately. And everybody probably in the room has heard of Chu and Fasman and Robson and Garnier. They produced these prediction algorithms that came to be about 60 or 70% correct. And many people, the beginning of structural bioinformatics, worked on different versions to try and improve, and they only went up very slowly. In fact, the main improvement came, and Chris Sander with Kabsch again developed the DSSP algorithm, and that algorithm just took a structure and defined where the secondary structure was, because there was a lot of disagreement. Where's the end of this helix? Is it residue I or residue I+2 or residue I-2? There was a lot of discussion about that. But with DSSP, everybody accepted that and moved on to the next step, basically. And but the prediction only really improved when Burkott-Ross in Chris Sanders' group included multiple sequences. And so you got a better prediction if you had multiple sequences rather than just a single sequence. And so that was kind of in the, I guess, in the 70s. In the 80s, we began to get more structures. I think, I'm not quite sure how many we had by 1980, but you could begin to look, instead of looking of completely disintegrating the thing, you could begin to see that the domains were really clear, and you could look at tertiary packing between the helices and the strands, and you can look at folds and architectures. And of course, all this time, we were getting many more sequences and many more structures. However, in the 1980s, horror of horror, it turned out that there were several incorrect structures deposited in the PDB. Now, structural biology is a very unusual part of biology in that if you do it properly, you don't make a mistake. If you do crystallography, you shouldn't make a mistake. And so this was quite horrifying for our crystallographic compatriots. And so the whole idea of trying to improve protein structure validation, looking at the stereochemistry of the local interactions. Can people in the room hear me? Yes, good. Let's try and carry on. So the validation, as I say, came out from trying to understand this interaction between the stereochemistry and the covalent structure and the non-covalent structure. And we got some wonderful correlations with resolution, better than you ever get in biology, because of course this is a human thing, so that our phi-psi values correlated very well with how good the resolution of the structure was. So this was a good way to look at the stereochemistry of the chain. And I should say, because I've not got a slide on this, Roland then did this as well and produced the side chain Chi-1 bins that have been really important in producing high quality structures since. That's Roland Dumbrack sitting on the front row here. But gradually, the importance of evolution came to the fore. And there've been lots of tools, obviously, to look at phylogeny. And we heard from Tanny Warno. And there's lots of tools for looking at multiple sequence alignments and for generating phylogenies. Also, with the sequence data, the start of the PFAM, Interpro, and SMART databases that really captured the different types of domains that we see. But perhaps the most important paper, I think, was that from Chothier and Lesk, who showed how the structure was really well conserved during evolution, and that even down to about 40% or below, we could see relationships. And you saw some in the first talk from Martin Steiniger. And this conservation of structure is what makes it possible to see farther back in evolution than from just sequence data. And of course, there were studies on individual proteins. In particular, I'd like to mention Anna Tramontano, who's been very important here for the immunoglobulin analysis with Arthur and Cyrus. And so the idea came about that maybe we could, instead of predicting the structure, we could recognize folds. And my experience of this, just to make it personal, I had a new student, Mark Swindells, and he was with Pfizer, and they wanted the structure of interleukin-1 beta. The structure had been published, but the coordinates weren't available. But this fold was exactly the same of that of soybean trypsin inhibitor, for which we did have a structure. So I sent Mark away in his first week of his PhD and said, go and align these sequences, then we'll build a model, homology modeling, and we'll have a structure for interleukin-1 beta. And he came back very shamefaced about a week later and said, I can't align these sequences. They're only 9.5% identical. How do I align these to get the structure? And I think at that point, I realized how far sequence could change and still retain the structure. It was quite, it wasn't very nice for Mark because he couldn't do that. So we were a bit stuck about interleukin. But at that time, there were many programs written to compare sequences, first of all, and of course, Jeff Barton's tool, JALView, to visualize these sequences, but also many programs to compare structures. And I actually wrote a program that I called Super, for superpose, to superpose two structures. So then I had Super A, because that was the next version, And then I had Super B, that was the next version after that. And I was quite horrified when somebody came to me and said, oh, your program's superb. It's really good at aligning these sequences, but in fact, it wasn't good. It wasn't anything like as good as the Saturn, Dali, and TM align. But there was this ability to compare structures meant that we could, because it was really difficult, 500 structures. And I couldn't remember them all. I mean, Alexei Mertzin, of course, knew them all inside out, but I certainly couldn't remember them all. And so having a tool, and this was why we started with Christine. So Willie and Willie Taylor and Christina Rengo developed the SAP algorithm. And that was the basis of doing the CATH data resource where we classified folds into different types. And you've seen this morning the brilliant work from Steiniger. So, and again, so a big problem was, right, you conserve the structure, but what about the function? And this slide that is years old now, from Christine, showed, who was there my postdoc, showed 4 structures whose sequence similarity in white were all below 10%. So they were very distant sequence similarity. But the core of their structures was the same, but their function, so as a non-biologist, the aspartate aminotransferase doesn't sound anything like a tyrosine phenolase. And so understanding that these structures could not only evolve, they conserved the structures, but they developed new functions. And again, this was quite an eye-opener for me. And the structures that in green, the similarity in green, shows that they're really quite alike. And those green numbers came out of the SAP algorithm. At this point, the term bioinformatics started to appear. So for the first 23 years of my research life, I wasn't doing bioinformatics because it wasn't an invented term. And of course, the sequence people all think they invented bioinformatics. And so it's an interesting, usual thing between different parts of the community. But with these abilities to compare structures, and I think we had about 500 in our first version of CATH, we could identify the all alpha ones, the all beta ones, and the mixtures alpha betas, which were the dominant ones. And we developed our classification. And at the same time, Cyrus Chothier and Alexei Mertzin developed the SCOP database. And it's been very interesting. Christine has worked very hard since to compare and contrast where they're the same. We were more algorithmic than Mertzin would just look at these structures and say, oh, those are related. I'm afraid we couldn't do that. So we had to use the computational way of doing that. But there were many software tools developed at that time in the 90s that went from full dynamics that was first proposed in the late 70s, electrostatic calculations, I think that Barry Honig talked about yesterday. There was a lot of ligand docking, which mainly actually took place in industry because they had the data needed to test that. There was the idea of reverse Boltzmann calculations of energy potentials from Manfred Sippel that was sort of a forerunner in a way of some of the AI, of the way that AI works in my mind. And of course, there was the covariance of close amino acids that Alfonso was involved with, but it happened, we'd all seen it, we all knew it was there, but when you tried to use it for prediction, it just didn't work. So that was the beginning. But there was fold recognition and threading where we tried to thread, from David Jones here, thread a sequence onto a structure. And of course, John developed the CASP thing because, so my simple interpretation of this was that several papers came out that claimed to have solved the protein folding problem. And of course, they hadn't. And so what we needed was this blind prediction, really influential in the field, I think, and as a forerunner of other things. But at that time in the 90s, there was a lot of prediction by homology modeling, and also the development of very good software tools for modeling the structure of a relative where you already know the structure. And I just have shown pictures here of Thorsten Schweder and Mike Sternberg, who have developed two of these tools that are widely used. They've both been made fellows this year of ISCB. So that's really rather nice. They've been acknowledged for what they've done. At the same time, there was these structural genomics projects, and those for probably 5 or 10 years were really important. The US, well, you can see the US invested a lot in different centers, but in fact, they were all over the world. And the idea, I think, came from Chris Sander to do the equivalent of the genome, but for protein structures. That was optimistic, I think, and proved optimistic. And of course, the problem was that we got these structures and then we didn't know what they did. And it was really hard, and is really hard, to tell from structure exactly what the biological function of a protein is. But it led to us beginning to look at the idea of going from structure to function. Then in the 90s, at the same time as this was happening, people began to look at the interactions of proteins, including protein-protein, protein-ligand, protein-DNA, and RNA. And we looked at all of those, and it was amazing how little data there was. We were lucky if we had 50 examples in the PDB that were unique, if you like. But since then, of course, these are now all captured in the string database that Per and many colleagues have developed. There was also this idea of function and capturing it. And I just wanted to acknowledge Michael Ashburner, who's no longer with us, for developing the Go tool. I can remember when he told me he was going to do this, and I was, frankly, a little horrified because I didn't think it was possible. Now, Michael knew an awful lot more biology than I knew, and he developed this tool. It's annotations. They're not perfect, but they have driven, actually, the field of function and function prediction. There's a long way to go, but I think Michael and many colleagues, again, all over the world, have been responsible. So then in the 2000s, I think the focus moved to networks, data integration, many more structures, the setting up of the comparative assessment of protein. Actually, I don't know what the RI stands for. CAPRI was docking and protein-protein interactions. We found out 30% of proteins in the human genome are probably disordered. And also the idea of predicting function from structure became real, and there was again the setting up this comparative assessment based on blind prediction, as Casper and John Mott had done. There was a lot of work on protein-ligand interactions, and again, just to give you a flavor, I thought this would be easy because the ligands, the small molecules, are so small compared with the proteins. What I discovered was that most of the software for the ligands wasn't available because it had been developed in the companies, it wasn't like bioinformatics where everything was open source, basically. And so we had to start looking, what is it? How many metabolites are there? I can imagine for several weeks we were discussing how many metabolites are there, and how do the metabolites compare with the drugs, etc. And just to mention Rafi here, there were lots of studies of binding in the PDB, which he was involved in, defining clefts and binding sites, comparing ligand conformation and promiscuity, which is a really important facet of many proteins in their binding. And the docking studies in Capri that Shoshana and others led, again, have been important. So this was look and see. If we look at these ligands that occur in many different proteins, what's the shape of the binding pocket? And what this shows is that different proteins create pockets that are different shapes. So just looking for pockets that look similar can actually not always give you the answer that you're looking for. And so there are many ways to bind molecules, many ways to bind molecules. So this was ongoing and people were doing it. But of course, the big change happened with AlphaFold and DeepMind and the setting up of the AlphaFold database at EMBL-EBI of these now 214 million structures. And the impact this has had has been quite phenomenal. So it's interesting because many of those structures could have been predicted by homology modeling. We know that already, but the quality, the accuracy of the DeepMind structures, I think, since it was published, has continued to surprise everybody. There are aspects that aren't working very well, but there are many aspects that are quite staggering. And this is like John talked yesterday, John Malt, about the folding pathway. And at one stage, I thought, we'll never predict the structure until we know the folding pathway. I quickly thought that's not right because there have to be many pathways because we know how flexible unfolded proteins are. And so to have this come about, and not only what amazed me with the AlphaFold structures was that they didn't have the ligands bound. And I still find that really challenging because we know if a zinc isn't there in a zinc finger, the thing will fall apart. But what they're doing is just learning the structure that's most likely to be found in the PDB. So it's a very directed thing. What it doesn't do is kind of explain to us, the bit that I enjoyed was always saying, type 1 beta turn, that occurs in a beta hairpin because of the twist of the sheet. So you could understand why you have these preferences. You can't do that. But the impact of this, the citation rate of the AlphaFold papers and the use of the AlphaFold database has been enormous. So I think as a field, both the experimentalists and us computational biologists, it's actually a huge boost. It's been phenomenal, I think, seeing the impact that it's had. And so what can we learn from structural data? And of course, the main thing for a structure is to shed light on the function or the mechanism, either to look at ligand binding or multimers or variants or whatever. And that's what most biologists will use these structures for. But as I said, when we, in the early 90s, I moved from Birkbeck to UCL and I decided, right, we're going to look at functions. So I thought, fold functions. What you rapidly discover is that the fold functions, that one fold does not equal one function because of evolution, quite the opposite. And so although these were the enzyme classes, and you can see they all have all types of structures in them. So that was a good start. We didn't get anywhere by doing that analysis. And so we gradually began to look at enzymes. The reason for that was that actually about 50% of the PDB is enzymes. And we have the EC classification, which is a better definition of function than for any other sort of protein in a way. But it's complicated. And so we started on the idea of trying to do computational enzymology, catalysis in the computer in some way. And we've done, we've done lots of different things about that. We have a database, we look at the evolution, we look at the families, we've looked at reactions and transformations, and we've also looked at mechanisms. By doing this high-level analysis, and this is work from a postdoc, Nick Furnham, who's now at London School of Hygiene, when we were looking at changes of enzyme families, we found that the vast majority of changes that occurred during evolution was to just change the substrate. Only a small fraction actually changes the chemistry, the catalytic reaction that happens. And so that was, to me, that was I know that protein enzymologists will say, of course, Janet, that's obvious. But actually to have looked at it and to find the data. Not only that, we found that the different colors are the different EC classes. And we found that there were examples already in the database where an oxidoreductase can change to each of the other 5 classes of enzymes. Evolution is amazing with what it can achieve. We've also created a library of 3D active site templates. This is Yanis Riziotis, a student who's just graduated. And so we now have this template library. But perhaps most exciting, and the thing that's been most challenging, is to try and do a knowledge-based prediction of enzyme mechanisms. So when I started, because I don't have any chemistry, I didn't do any chemistry. So the last thing I should be doing is looking at enzyme mechanisms, because it's real hardcore chemistry. But I really felt that, proteins are all the same. They're all polypeptides. So our catalytic toolkit in a protein is very limited. We've got the 20 amino acids, of which only 6 or 7, the polar ones, are involved in catalysis, and the cofactors. So the catalytic toolkit, it's like going into the garage and getting out the tools, is very limited. And so it seemed to me that with all of the knowledge we now have about mechanisms, it might be possible to use bioinformatics to predict mechanisms. And Antonio Ribeiro has been working on that as a real functional thing. And I think there's a lot more work to do on that. It's published, it's in Nature Methods last year. But it's the beginning of being able to predict catalysis. And I think that's going to be important when we're trying to design new enzymes with new functions. But I wanted to close, well, not quite close, but so what's the most exciting thing? So remember, I started out with just 20 structures. And every week when Nature and Science came out, we'd all go and look for a new structure. What did it look like? We had no idea. Now, of course, we've got 214 million structures. So the question is, well, what does this universe of protein structures look like? And I just want to mention the TED database or encyclopedia of domains and say right at the beginning, this isn't my work, this is from Christina Rengo and David Jones's group, and Nicola Borden will be speaking later on in this session, I think, about this. So I won't go into details, but I just want to show you. So we've got 214 million AlphaFold structures. You can compare them. Are they new or are they old? And in order to compare them, you have to compare with what we know. And so, of course, Kath, this is sort of, to me, the proof of the value of doing Kath, because you can compare these structures against the ones that we already know. And they found 7,500 new folds. And I just want to show you these. These are symmetric folds. I hope Nicola doesn't mind me showing these because they are so beautiful. I mean, I think I got into protein structures because they are so beautiful compared with a linear DNA sequence. You know, you can see them. Well, they are. And you can see these amazing symmetric structures, which are just like flowers. And of course, you can also see, and this is a bit complicated, so Nicola, I'm sure, will explain it, but that certain families have expanded. And in fact, Martin mentioned some of this in his talk this morning. The universe of protein structures is dominated by a small number of folds that have been duplicated and duplicated and do many, many different functions. And so this is, you know, this is really exciting, I think. But it's not the only exciting thing. So where is this all going? So for the experimentalists, there is a new normal. Now, when they're starting to solve a structure, they will look into the AlphaFold database and say, oh, is there a structure there? And if it's an EM structure, they will perhaps use that structure as the model to identify it. So it's changed the way that structural biologists, the experimentalists, think. And so in a way, this is Samir's slide, another golden age for structural biology, and I would say for structural bioinformatics as well, because the ability to model, this is the nuclear pore, and it was modeled by Jan Ellenberg at Embl, Hamburg. And you can see that with the cryo-EM of cell extracts, you can build these models of very complicated structures. But not just that, we can begin to look into cells and see molecules because the resolution is high enough to see large complexes. And so we can begin to look at the architecture of the whole cell. And with the human cell atlas, when we get the different types of cells, if we can get these pictures and be able to look at these and then identify, the imaging techniques today are phenomenal. So here we can see a slice through a tomogram, and you can you can recognize this as a chaperone. You can see the ATP synthasers, and you can see this structure, which they didn't know what it was. They've actually looked and they've done some experiments and they think they know the 4 proteins that create this something. And so with this cryo-EM and the tomography, the ability to look is fantastic. And this is 3D biology. Perhaps it isn't just the 3D of the proteins, but it's the 3D of life. It's what happens in life. And of course, whilst this is going on, when I joined, I didn't mention, when I went to EBI as director, there were 5 databases. We now have over 41 data resources. And that's in 20 years. So they go all the way, of course, from the genomes down to the systems, and we have many different resources. And in my world, the protein sequences and structures are at the heart of the world because they're the things that do things, they're the functional things. But of course, there's all the other biomolecular data and the multi-omic. There were many, many posters about multi-omic data analysis. But perhaps even more exciting is the integration with the medical data resources, which have yet to emerge. The sharing of medical data is a real challenge. The biodiversity, the natural history museums around Europe are all getting together to digitize the data on the fossils and the dinosaurs and everything. So you'll be able to use that data as well to look back and to create it. And of course, the chemical data, because the chemistry of life is quite important. So in my mind, structural biology is one part. Of course, it's the best part. We know it's the best part. But, you know, this is one part of this bigger field. And even today, there is clear silos between these different disciplines. But the only way to bring this together is really the computational biology way. And so, as far as I can see, the future is very bright for structural bioinformatics. So this slide is put together from, I give a talk about structural bioinformatics, and these were suggestions from the people in the room about the sorts of things. I'm not going to go through all of them. You can see them. Perhaps the most exciting in my mind is the protein design and using these things to really design new molecules that can do new processes. So I'd just like to finish and acknowledge the people, my group that worked on the enzymes. I'd like to acknowledge Christine, who I've worked with forever and has been a wonderful scientific companion, and David, who, well, is just very good. Not that Christine isn't quite there, but it's different. People think differently, and that's one of the joys of science, that you have in your group people who think differently, and then you bring them together, and the synergy is fantastic. And Samir, of course, for heading up now the Protein Data Bank in Europe at the EBI, but also to the Worldwide Protein Data Bank that's been so essential for all of this. So last December, we had a get-together of all the people, well, not all, but a lot of the people who'd been in my group. And it was wonderful to see everybody and see how they'd all developed and grown, and still most of them doing something scientific in different fields, though. And so I think bioinformatics training gives people the training necessary to go on to really great things. So with that, I will end.
Thank you so much, Janet. It's very touching and very inspiring at the same time. Because of the technical problems, we will have one or two questions. I see one here. So.
Hello. I was wondering about the enzyme substrates that you were looking at. Did you also get to consider transition states, which might not necessarily be represented by observable molecules, but nonetheless have some impact on the shapes, perhaps?
I didn't go into anything on the detail. We can talk about this later, but it was, to me, it was such an interesting project because there was no database of enzyme mechanisms. It was just pictures. So we had to define how we represented that mechanism and the different steps that all came from the literature, and then capture it with cheminformatics tools, and then do a search for what might happen based on the most likely steps that you see in all the mechanisms brought together. So it's to me, it's still a fascinating problem. But as I say, I wasn't really the person to do this. I'd avoided it for 10 years. But I think it's fundamental because it's all about how enzymes do catalysis, which I think will be important for designing new ones.
Any more questions for Janet?
No, we can.
If no, let's thank Janet again.
Can I also apologize to those people who I didn't explicitly mention or whose work I didn't mention, because I realized this morning, I thought this is impossible, totally impossible to do this, to do justice to it. So thank you to anybody who I didn't mention, but who's made a big contribution. Thank you.